Obesity is one of the biggest public health challenges facing not just America, but every industrialized nation. So, doctors around the globe are aggressively pursuing new experimental treatments to stem the rise of the obesity epidemic. In medical school, no doubt you'll study some of these new future treatments. But of course, understanding that science means understanding basics about lipid metabolism by the time you get to medical school. I'm Tyler Farah, and let's study lipid metabolism together right now. In this lesson, you'll see how fundamental science about energy density for sugars and fats and about lipid and sugar metabolism can be exploited by modern experimental research techniques. Take a moment and read through this question stem right now. Wow, what a question stem. This one is going to require a little bit of unpacking. We're told that this thermogenin protein, quote, uncouples the electron transport chain from oxidative phosphorylation. Well, we've seen this before. In fact, we just saw this idea in the aerobic metabolism lesson. There, it was a molecule called 2,4-dinitrophenol that did the uncoupling. But recall the effect this had on the cell. The cell would burn fat and sugar to make a proton gradient in the mitochondria. But instead of using that proton gradient to do something useful like make ATP, 2,4-dinitrophenol dissipated the proton gradient before those protons could be used to make ATP. So uncoupling the ETC and oxidative phosphorylation means you're burning fat and sugar like normal, but you're wasting that energy as heat. The maybe surprising thing about this question stem is that apparently your body has a gene that you used to express that makes a protein called thermogenin, which your body used naturally for this same purpose. So I want to check with you. Actually, almost every cell in your body has the same set of genes. That is, chief cells in your gastric pits have squamous epithelial cell genes, and squamous epithelial cells have helper T cell genes, and so on. So, how does a cell like a chief cell become a chief cell and not a different cell type? The answer is differential transcription. Your body can turn some genes on and other genes off, and this process of regulation is constantly occurring in the cells of your body. So it is with this thermogenin protein. The fact that you ever once in your life made the thermogenin protein means that the thermogenin gene is still in the cells of your body. And that's where these researchers' strategy comes into play. They want to use this sophisticated mechanism to turn the thermogenin gene back on, thereby causing weight loss. The rest of this lesson is all about exploring the mechanism they've proposed, which is going to mean combining MCAT-level molecular genetics, biotechnology, and metabolism. To start with, the suppressor protein is a kind of transcription factor. So by what mechanism would a suppressor protein suppress a gene like the one for thermogenin? Transcription factors like this suppressor protein work by binding onto DNA and changing the way it gets transcribed. In this case, the suppressor protein binds on and blocks transcription, so the thermogenin gene never gets expressed. And that means that the thermogenin protein never gets made. But here's an interesting question. The suppressor protein is itself a protein being made in your body. So where does the suppressor protein itself come from? Well, the suppressor protein, like all proteins, must have started as a gene, and that gene then got transcribed into mRNA. That mRNA exited the nucleus and bound onto a ribosome, and that ribosome then translated that gene into the actual suppressor protein, the suppressor protein then going on to block another gene. So the scientists in this question, their goal is to block the suppressor protein, which then turns the thermogenin gene back on. Scientists have a cool fancy word for mechanisms like this where you're suppressing the suppressor, and that word is disinhibition. But basically what we're told is that microRNAs, which are small chunks of RNA, are going to be used to suppress the suppressor. What we're learning, though, is that there are actually several possible targets for these microRNAs in this overall mechanism. These microRNAs might be used to attack the actual suppressor gene on DNA itself. The microRNAs could potentially target mRNA transcripts made from that gene, or if we're looking to shut down a protein, another possible target is the protein itself. My question for you is, of these three possible targets, the DNA, the mRNA, or the protein itself, which one seems like the most likely target for microRNAs and why? The answer is that microRNAs will target the mRNA transcript of the suppressor gene. The microRNAs really have no way to attack the suppressor protein itself. There just isn't any way for a small chunk of RNA to disrupt a stable, fully formed protein. Similarly, the microRNAs can't really attack the gene on DNA because DNA is double-stranded. Pieces of genetic material generally can't bind onto DNA because it's protected in this way. And in fact, this kind of protection against attack by polynucleotides is one reason why DNA evolved to be double-stranded in the first place. By contrast, mRNA is single-stranded, which means a piece of complementary microRNA can bind right on to the exposed nucleotides of the mRNA molecule. My next question for you, why will a microRNA binding onto the mRNA transcript block translation of that mRNA transcript? Ribosomes are responsible for translating mRNA, and ribosomes rely on mRNA being single-stranded. The ribosome latches on to a single-stranded mRNA molecule, then matches chunks of tRNA, which carry amino acids, to the exposed mRNA. So if we bind up the mRNA to microRNA instead, then the mRNA cannot effectively bind to the ribosome. In fact, in recent years, scientists have discovered dozens of these microRNA molecules that are active inside your own body naturally right now, suppressing gene expression in this very way. And some researchers, like the ones described in this question, are actively researching treatments based on microRNAs. As a doctor, there's a very real possibility that as you're starting your career, some of these treatments will just be coming available on the market. In any case, this has been a complex mechanism, so before we move on, put it all together for me. How exactly would the microRNA molecules described in this question cause weight loss? In summary, microRNA suppresses the suppressor for thermogenin. This disinhibition of the thermogenin gene means that the thermogenin protein will now be expressed in an adult. The thermogenin protein then causes weight loss by uncoupling the ETC from the oxidative phosphorylation process. This means the cell burns fat and sugar, but doesn't actually make any ATP, so that energy is just wasted instead. This leads directly to the next prompt question. Our researchers are worried about where in the body they activate the thermogenin gene. We're told that they want to avoid activating the thermogenin gene in tissues that rely primarily on carbohydrates for energy. They really only want to turn thermogenin on in adipose tissue. Our goal is to figure out why this is so important. And to do that, we're comparing glucose and decanoic acid. Decanoic acid is a 10-carbon saturated fat. It is stored in limited amounts in your body, but decanoic acid will also be an intermediate of the beta oxidation of any longer chain saturated fatty acid, such as, for example, the more common palmitic acid. So this is a biologically relevant fact. And the reason why it's a good comparison to glucose is that glucose and decanoic acid have similar molecular weights. So speaking of oxidation of biomolecules, during metabolism, fats and sugars are fully oxidized. Why do we oxidize fats and sugars? And what does fully oxidized carbon end up looking like? Recall that oxidation is loss of electrons. And in metabolism, those electrons, of course, go on to the electron transport chain. Often, especially in biochemistry, when an atom is oxidized, that atom physically binds to oxygen, which is actually where the term oxidation originated from. So fully oxidized carbon is CO2, carbon dioxide. In fact, the carbon dioxide produced by metabolism is the very same carbon dioxide you force out of your body each time that you exhale. So here's another question. Even just looking at sugar, why would sugar be somewhat less useful for metabolism compared to fat? Why would sugar be less oxidizable? The answer is that the carbons of sugar are already partially oxidized. Each of these carbons already has at least one bond to oxygen. So the carbons of sugar have already surrendered some of their electron density. Since the goal of sugar and fat metabolism is to generate electrons for the electron transport chain, sugar just contributes fewer electrons carbon per carbon compared to fat. Additionally, these molecules differ in the number of carbon atoms in the first place. Why is that a significant observation? The endgame for sugar and fat is CO2, the electrons extracted from carbon along the way being used in the electron transport chain. So glucose with 6 carbons simply has fewer carbons available for oxidation. Overall, gram for gram, sugar has fewer oxidizable carbons, and then also the carbons that sugar does have are already partially oxidized. So scientists say sugar is less energy dense than fat. So another question for you. If sugar is gram for gram less effective at storing energy than fat, what are some advantages of sugar over fat? Why does our body bother with sugar at all? For one thing, sugar is available in the environment, so it makes sense that our body would have a mechanism for metabolizing sugar directly. But moreover, sugar is water soluble, fat is not. And think about it, every part of using fat is inconvenient for your body. You need a special mechanism involving chyme to digest fat in the first place. Then you need another special mechanism with chylomicrons in the lymph system for transporting fat from the GI to the liver. Then another special mechanism involving lipoproteins to transport fat from the liver to the rest of the body. And lastly, you need specialized adipose cells just for storing fat. All of this special treatment boils down to the simple fact that fat is not water soluble, so it needs that special treatment within your body. In short, the advantage of fat is that it has such high energy density, but the advantage of sugar is that it's much easier to use. And that leads me to my final question. If a cell relies primarily on sugar for energy and you turned thermogenin on in that cell, why would that be dangerous for that particular cell? The answer is that the cell would rapidly run out of sugar to burn. And this is because sugar is less energy dense than fat. In fact, glycogen stores in your muscle, for example, can be naturally depleted after just a few hours of low intensity exercise. Now throw thermogenin in the mix, which will be wasting that energy instead of using it, and you can see why that would be a problem. This is in fact the hurdle that researchers currently face with this whole mechanism. They have the ability to make microRNAs that could turn thermogenin back on in your body. However, currently researchers haven't figured out a mechanism for targeting adipose tissue specifically. Lessons like this one are a great reminder that the cutting edge of modern medicine is not so far away. The most complex, sophisticated, or experimental techniques still have their underpinnings in the fundamentals you're learning for MCAT science. I hope that's an encouraging message for you as you continue your press up to test day.